One of the things Mendel's work did not take into account, basically because he didn't know that they existed, is the fact that chromosomes, the 23 pair you have, the four pair that fruit flies have, however many pairs pea plants, forget how many that is now, that those chromosomes are literally nothing more than hundreds to thousands of genes linked one after another. Mendel's work is based on one character, two alleles. Different characters not interacting, each having two alleles, two traits. In hindsight, looking back from the 21st century back to the 19th century, when Mendel was doing his work, we see, as I have made the joke many times, somebody upstairs was looking down and, blessing Father Mendel, because it turned out the characters he chose to study all appeared on different chromosomes. Fast forward decades later, as we begin to study not only pea plants again, but other organisms, we see that independent segregation and independent assortment does not apply. Because we start to find that where one character goes, one gene goes, others go along with it. So we came up with this idea of linked genes. Genes that are linked together because they are literally on the same chromosome. Morgan, he of the red eye versus white eye experiment. Morgan, during his characterization of fruit flies and his breeding experiments, him in his lab, and I say his lab because you got to remember, it's not Morgan in the lab by himself doing stuff. He has grad students, he has undergrad workers. His lab were doing breeding experiments and seeing how traits were passed on. He noticed that certain traits, certain wild type phenotypes, certain mutant phenotypes just seem to be passed on together. Where one went, the other went. Peanut butter and jelly, they just seem to go together. Great example of this is black body and vestigial wing. Remember I told you one of the mutations that Morgan talked about in his research was black bodies. What does that mean? Well, the wild type, B+, means that these bodies were tan with black stripes. B, these bodies were all black, no tan present. VG was a mutation that gave vestigial wings. VG plus were the wild types, the wings extended out beyond the length of the body. VG, these were the vestigial, the little nubs that just sat there and just kind of fluttered as the fruit fly walked around. Morgan and his lab developed their version of a test cross. Remember, Mendel came up with the idea of test cross. If you're not sure if something is homozygous, heterozygous, cross it, hybridize it with, a known recessive. See what the offspring is. Well, here, Morgan and his lab didn't know if black and vestigial wings were somehow linked together. Somehow where one went, the other had to go. So they did a test cross. They took what they theorized to be a heterozygous, double heterozygous, and crossed it with a mutant, black-bodied vestigial winged fruit fly. Then they wanted to see what would happen. Are all of them going to be normal? Are all of them going to be the mutant? Or are we going, is it going to be 50-50? Just like you started with? Or are you going to get some form of ratio? 9-3-3-1 like Mendel, or some weird different computations of. So let's look at this. B plus B plus, VG plus VG plus. Slightly different than what was the last chapter, but this is what Morgan initially did. This would be post the red-eye, white-eyed experiment after it was published. So, double wild type versus BB VG VG, which is a double mutant, black-bodied, and notice those little nubs right there, vestigial wings. This is a double wild type. This is a double mutant. Both true breeding. Boom. Here is what we talked about last slide. B plus B, VG plus VG. The F1, which is a dihybrid. Okay, a dihybrid wild type, heterozygous for both genes. Now we want to see what happens. We want to see if where B goes, does VG also have to go? Where the B plus goes, does the VG plus also have to go? Will you end up with the two parentals all over again? So they did the cross. BVG, because that's all this can do. And then remember, as we talked about last chapter, B plus VG plus, boom. B plus VG, boom. BVG plus, and then BVG. There you go. Boom, boom. So you have the 4 different types of gametes. If wherever B plus goes, VG plus has to go, then all you're going to get is wild type and this, black vestigial again. These two will not occur. But Morgan theorized that these two genes are linked. Something about where B goes, VG has to go. Something about where B plus goes, VG plus has to go. If it was true Mendelian and everything was on different chromosomes, it would be a 1-to-1 to 1 to 1, okay? Because everything's 50-50. If they were truly linked, where B goes, VG has to go, where B plus goes, VG plus has to go, it'll be like I said, 1-1-0-0. But, and here's the big but, Morgan was able to show that they would get all 4, but not in the 1-to-1. They didn't get the 1-1-0-0. What they got, 5-5-1-1. Definitely not a Mendelian ratio. They tried this with other what they thought of as linked genes, and the numbers were not the same. The ratios would be slightly different, but they would always end up with some small percentage that were not the parental phenotype. Seemed to be a mix somewhere between the two. We now know this as genetic recombination. We talked about this in the meiosis chapter during prophase 1 when all the chromosomes line up to form the tetrad. Homologous chromosomes can get entangled and the different parts will be fixed and you'll get swapping. Remember, red chromosome with blue tips, blue chromosome, red tips. So that genetic recombination explains the 5511. Recombination, as I said during the meiosis chapter, can occur, but it doesn't always.